When we are studying the regulation of gene expression, lack operon is a very good example to explain the regulation or how the gene expressions are regulated. Before we start with the lack operon, first understand what is an operon. The concept of operon was given by Francis Chekhov and Monod. Now, they said that all the genes controlling a metabolic pathway as a unit constitute an operon. So, according to them, the genes which are regulating a metabolic pathway, all genes that regulate metabolic pathway will form the part of an operon, and there can be many operons. So for example, the E. coli lack operon we have, then we have TRP operon, ARA operon, VAL operon. These are various kinds of operons, and all the genes are taking part in this metabolic pathway. When we have to study the components of an operon, when we talk of various components, there are certain components of operon. The first is the structural gene. So the first component over here in this case is the structural gene. The structural gene have the information to produce the enzymes, and they are transcribed, and that is into mRNA. The second is the operator. Operator gene has to be present, and this operator gene is a sequence which lies next to the structural gene, and it regulates the expression of the structural gene. That is, the action of the operator is regulated by proteins, and these proteins are called as repressors. Then the third part is we have the promoters. These are various components we are talking of. So the promoter gene is located next to the operator. So we have the structural gene, and structural gene has closely associated operator gene, and the promoter is closely to the operator. Now this operator, it has two functions. Sorry, the promoter has two functions. The two functions are, since it has the sequence of bases where the RNA polymerase binds for transcription. So the RNA polymerase binds over here, binding of RNA polymerase. It happens because the sequence is in a way that the binding takes place easily. And secondly, it has a base sequence that decides that which out of the two strands of DNA, which will function as the template strand. So the promoter decides that first it helps in binding of the RNA polymerase, and secondly, which DNA strand will undergo the process of transcription. The next is the regulatory gene. And this regulatory gene codes for a protein which is called as repressor. So it is repressor gene. And when this repressor binds to the operator, that means when the repressor gene binds with operator, the operon is switched off and the transcription does not occur. If the repressor is unable to bind to the operator, so what happens is this repressor if it binds to operator, no transcription will take place. Since we are talking of lac operon or in terms of this. And when this repressor is not binding to the operator, then the switch is on and the transcription continues. So over here it is off mode and over here it is the on mode. The last we have is the inducer. The inducer is a metabolite that binds to the repressor. So what happens is, the repressor is basically, if you see over here, the repressor, if it is binding, then it is not allowing the transcription to happen. If it is not binding to the operator, then the transcription process continues. So the inducer if it binds with repressor, changes its configuration and it prevents the repressor from binding to the operator. So inducer ka function, what is the function of the inducer? Inducer will bind to the repressor. It will change its configuration in such a way so that it is unable to bind to the operator, and therefore it will become non-functional and the process of transcription takes place. That is, it happens in the case of lac operon, where lactose is the inducer. So what is happening over here? This will change the configuration of the regulatory gene or the repressor, and as a result the repressor will now no longer be able to bind to the operator. If it is not binding to the operator, then the transcription is on. That means the synthesis will start. Now, coming to the lac operon, which we are more interested, lac operon was again discovered by Francis Jacob and Jacques Monod. Now these two scientists explained about the lac operon mechanism. Now if the lac operon, it is an inducible operon. Why are we calling it as an inducible operon? Because here lactose is the inducer. That means it regulates or inactivates the repressor. If the lactose is present over there, then it will bind to the repressor and it will not allow it to in turn bind with the operator, and therefore the transcription will take place. And therefore, if we study about the lac operon, it has following components. Now, structural genes. There are 3 structural genes over here, and they are linked and they lie close to each other. The 3 structural genes are that is, if we talk of the structural genes, there are 3 structural genes over here. The first is the Z gene. Now Z gene codes for enzyme beta galactosidase. It codes for beta galactosidase. And it catalyzes the breakdown of lactose into glucose and galactose. Now this glucose and galactose are utilized by the bacteria, that is E. coli. We are talking of lac operon in the case of E. coli bacteria. So in E. coli, if Z gene is there, then beta galactosidase is an enzyme that breaks down the lactose into glucose and galactose, and which is then utilized by the E. coli for it is as energy source for these products are used as an energy source by the E. coli to perform its various metabolic activities. The other is, this is a beta galactosidase enzyme. Then next is we have the Y gene. The Y gene is coding for an enzyme permease. So this gene codes for an enzyme permease. This permease enzyme increases the permeability of the cell to beta galactosidase and lactose. So this Y enzyme, that is permease, you can remember by the name since it allows permeability against whom beta galactosidase and also for the lactose. Then the last third structural gene is the A gene. You can see on the extreme right hand side. This is for trans-acetylase. So this gene codes for trans-acetylase. Trans-acetylase enzyme, and it makes the lactose into its active form for metabolism. So it activates the lactose. Now all the three gene products are required simultaneously for the metabolism of lactose. So all these three structural genes are to be present for metabolism of lactose. All these are required. Now there is these genes form these genes are necessary for the lactose metabolism. Then next is the operator gene. Next we are going to talk about the operator gene. The operator gene, when we try to understand in this like Operon, there is a single operator gene which controls all the three structural genes as a unit. And this operator gene over here is O. So O over here is the operator gene. Next is the promoter gene. So we have now the promoter gene. The promoter gene, it is actually the sequence of DNA which is close to the operator gene, and it provides a site where the RNA polymerase enzyme binds to start with the process of transcription. So over here the promoter gene is the P. Promoter gene over here is P. It is the site where the RNA polymerase enzyme will bind for the process of transcription. Then we have the regulatory gene. Regulatory gene codes for the repressor protein, and it is also represented as I gene. So I gene is the repressor protein. I gene is the repressor protein. Now it is inhibitor of Operon, that is when it binds to the operator. Then we have the inducer, and inducer over here is lactose. Lactose is the inducer, and therefore lac is the term we are using over here. That's why the lac operon or the lactose operon. Now over here you can see the inducer binds to the repressor and changes its configuration so that it cannot bind to the operator. Now what happens over here in the first step? The inducer is binding to the repressor and changes its configuration. So as a result, it cannot bind to the operator. Now the repressor and inducer complex move away from the operator, and the operator switch, that is in on mode. That means the synthesis will take place, and RNA polymerase has then the access to the promoter, and the transcription happens, and the lactose is catalyzed into glucose and galactose. Now, regulation of lac operon is the regulation of enzyme synthesis by the substrate lactose. And the regulation of this operon by repressor is referred to as negative regulation, whereas that by inducer lactose is a positive regulation. So what happens over here is that when the repressor binds, then the promoter will not be able to synthesize with the beta galactosidase, permease or trans-acetylase, and as a result this process or the gene expression will be inhibited or stopped. So we can say the regulation of lac operon is a regulation of enzyme synthesis by the substrate lactose.